So, good evening. Sorry, I think I'm a little bit late. I got caught up with some few issues, but I believe the next time we'll be a little bit punctual and prompt on time. Today we are going to be looking at microbiology. I believe you people have been covering various topics in this, and I'm just here to do a little punching up of the spaces that have been left. I know most of you understand what we mean by microbes. I know most of you understand by what we mean by microorganisms, and we have to put one thing into our minds right now, that this is a subject that is very diverse. Okay, yeah, it's because we are studying about the smallest thing that we can't even see with our naked eyes. That means the room for microbiology is extreme. Okay, you could develop in any field once you have the knowledge of microbiology. We have to understand that right now the world is focusing on those things that the earlier scientists didn't get chance to verify or find out because they didn't have the technologies that we do have right now. So today we will be looking at food spoilage, and in food spoilage we are saying this refers to food that has been damaged or injured, making it undesirable for human consumption. Really, no one wants to eat food that is spoiled, and we will see that there are various characteristics of food that is spoiled, and various causes of this spoilage. Really, this is something that I think we've all come across. We've all come across spoiled food. Once someone has been taking milk and they stop taking the milk, or they reserve that leftover milk, the next morning when they come back, that milk has gone bad. That's what we usually term it as, that food has gone bad. Uh, Meat has gone bad, fish has gone bad, the egg is bad or it is stale. Yeah, a lot of definitions that we really use when we are trying to define what food spoilage is. But really, that is what we can give you as a statement that will look at being damaged or injured or making it undesirable for human consumption. Well, this is also relative, friends. Like I said, undesirable for human consumption. Here we are looking at when we talk of undesirable for you as a particular person, maybe Clara, you might think what is undesirable to you is undesirable to me, but in actual sense, it is not. It is desirable to me. For example, some people like the texture of yogurt. Whenever they take it, or they want smoothies, but other people find that undesirable to them. So we need to use that one in caution. Let's look at the causes of food spoilage. And one, we are looking at microbial degradation. Because we are in a microbiology class, we will focus most of our efforts on this. And we are saying microbial degradation is a process in which these microorganisms, for example, the bacteria and the fungi, break down these materials. Okay, and also we are looking at that being the first cause. The bacteria, the fungi, and other spores and all those other things lead to the degradation. I'm getting feedback from someone. I think someone's mic is open. I don't know. Should we mute a little bit? Okay, that's good. The other thing is we are looking at rodents. When we ask for causes of food spoilage, I think you will be able to reproduce them. But really, according to the way I know this class, you people are a little more brilliant, and you can always come up with other things that are still in line. I don't want everyone trying to reproduce the work that we always give here in class. We want you people to go extra miles and discover for yourselves, because once any person in academics has not yet conducted any research on their own, that means they are in elementary school, or what we call primary. Yeah, but we expect you to be able to go and look for I think that is the time when they say that now when a man grows, they will leave their mother's home, their mother and father's home, and they will go and start their own family. So this is what we want you to do. We want you to leave what Risto Institute has given you. We want you to have the understanding of what Risto has given you, but I want you to look at the world beyond what we give you here in class, because we can never summarize everything that is there in the world in the small engagements we have. We are looking at rodents, and we are saying these create pathways for microorganisms by burrowing and spreading organic material. For example, these rats do droppings, and once they do these droppings attract the microorganisms that will come and lead to spoilage of our food. Insects, especially those feeding on organic matter, and we are saying physically break down materials and create entry points for other microorganisms to come and uh, inhabit that particular food. We're also looking at physical injury like bruising, freezing, which weakens the organic materials, making them more susceptible to microbial attack. For example, here we'll look at the fruits in detail, fruits and vegetables. In most cases, when people go to purchase fruits, they want those fruits that are not bruised. I am not so sure. Maybe you did some course unit on cookery. So if we did a course unit on cookery, then basically we looked at fruit selection. So with physical injury, really we see that now the dermis, okay, yeah, or the endocarp, I don't know, whatever kind of protective layer that is there for the fruit is now damaged. And once this is damaged, now the fruit is exposed to external factors. And one of the external factors are the microbes, free living microbes in the air. So they will use this opportunity and infiltrate that particular food. Also, we are looking at tissue enzyme activity, which continues to break down tissue even after the organism's death, fostering microbial growth. Yeah. So, the tissues, for example, when we look at the tissues that are found in meat, for example, structures like collagen, in case there is enzyme activity going on, remember the role of enzymes is to induce a reaction, and the end point of this reaction is to break down something, to break down a substrate into a product. I think we know that. So once this still goes on, we understand that these tissues are broken down into products, and in most cases we tend to know that these products, at the end of the day, more I think the microbes find it more appealing to associate themselves with these products that come out of the enzyme reactions. So we are saying that this could also foster a microbial growth, and non-enzymatic actions such as oxidation, hydrolysis also lead to a breakdown of these complex compounds into simpler ones. This could also able and aid microbial metabolism. Well, with microorganisms themselves, they are the primary agents of degradations. Okay, these some of them produce enzymes to break down the organic molecules that are crucial for recycling the organic matter in ecosystems and nutrient cycling. Well, if we ask for the causes of food spoilage, I think you could be able to list a few things around this slide. Let's proceed. Okay, so when we talk of microbial deterioration, this word always plays me. Sometimes I call it deterioration, then the other time it is deterioration. Yeah, but I think we get the concept. Deterioration is something going bad, or something coming from a state of good going to bad, or things becoming worse. For example, we could come and say that that man's health has deteriorated. That means that man's health is going, like he's having an illness that is giving him challenges with his health, and the immunity is deteriorating, which means it is going reducing, kind of something like that, but in a negative way. So when we talk of microbial deterioration, we are saying it encompasses various changes in the material, and the material here we are looking at that particular foodstuff that you have. For example, the appearance will change, and remember, when we're looking at food product development, we're looking at more of what are these factors that appeal to the consumer. For example, now if you're selling your fruits or you're selling a vegetable, this visual element or the visual aspect is more what opens the consumer to your particular product that you're trying to sell to them. So once their visual taste, once they visually detest your product, then they won't purchase it. So visual aspects may shift, leading to discoloration or spots or general decline in quality. That one comes from the microbial point of view. Once you have microbes in a certain food, then the appearance could change. Then you are also looking at the color. Okay, so we could maybe say it is kind of the same, but where we are looking at microbial activity altering the material's color, resulting into darkening, browning, or emergence of unusual colors, often indicating specific microbial involvement. And this we usually see it still in fruits. For example, the mangoes will start looking a little bit brown. You expect the mango to be yellow. Is it yellow or orange in color? Yeah, but then we want that mango to be looking a certain way. But once we start looking at a brown mango, then we get high on alert and say that maybe some deterioration is taking place. With the textures, we are saying the microbial degradation can affect texture, causing softening, wilting, and also development of slimy or mushy consistency. And this is dependent on the specific microorganisms and material type. Odor, also something that we could associate with smell. We are saying that microbial deterioration introduces altered and sometimes unpleasant and distinctive odors as microorganisms produce volatile compounds during decompositions. Friends, I think here we will need to differentiate. At some point, it's not that it is the work of the microorganism. It is not the microorganism that is giving off the flavor, but it is the activities that the microorganism is doing. Maybe its waste products, maybe its breakdown, maybe a lot of issues could come in handy around there. Yeah, and also we are looking at the flavor. We are saying microbial activity can change the taste of the material, which will introduce sour or rancid flavors. And I remember we looked at rancidity when we were looking at fats. I hope you remember something when we were doing product development. If you didn't see anything like rancidity or oxidation, then you need to go back and revisit those slides. Then finally, we are looking at certain microorganisms, especially the bacteria and molds. These generate visible slimy and mucilaginous substances during growth, impacting both appearance and texture. I don't want to disgust you, but when we talk of something slimy, the first thing that should come to your mind is the mucus. Okay, anything slimy, yeah, and most people don't want to associate themselves with such things that that consistency, the slimy consistency. Yeah. Okay. So I think that is that when we are looking at the microbial deterioration, we've looked at the causes first, where we have looked at things to do with the physical injury, things we've looked at to do with the tissues, we've looked at the non-enzymatic and the enzymatic, rodents, insects, etc. Then we've also looked at then the deterioration. How does it deteriorate in appearance, in color, in the texture, giving off odors and off flavors. And also we've looked at the slimy production that comes. So friends, now let's interest ourselves with the food classification and the spoilage categories. Well, here we are looking at the foods. Some of the first class we are going to look at the stable or non-perishable. Here, ideally, friends, we are looking at this particular food or this particular commodity being stable and perishable. That means it takes time to go bad. For example, we are looking at the flours, we are looking at the canned food, but with canned food, let's have some little precautions right there. Then dried pasta. Pasta, this is where we are looking at the macaroni, we are looking at, you're looking at, how do you call that? Spaghetti, yeah, etc. You're looking at rice, you're looking at sugar. We are not saying that these things don't go bad. Well, we understand that there are contaminants that could come in handy, but really it is once kept in, sometimes we always use that cool, it's a cool dry temperature, or cool store in a cool dry place, something like that. Once stored well, we will expect that these particular commodities will have a longer shelf life compared to the other commodities. So the characteristics we are looking at, these having a long shelf life and also resistant to spoilage and microbial growth. And we're also saying that they are often processed or packaged, and also they are stored in a way that prevents deterioration for extended periods of time. For example, these are things that we usually can export, or usually we can even keep if we are in business. These are things that we can keep because we know they're not going to get spoiled, so that we can sell them when the prices increase. So that's why we want to encourage most of our farmers not to sell their maize to middlemen, rather go and make maize flour out of it, so that you can keep that maize flour. Once you could keep that maize flour well, then maybe when prices go high, yeah, you could fetch some better income compared to what people were giving you earlier on when the season, when there was a lot of maize abundant during that harvesting period. Well, we are done with what we are looking at as a stable. Now let's interest ourselves with a semi-perishable. With a semi-perishable, we are looking at potatoes, apples, root vegetables, and some grains. And we are saying that these items have a moderate shelf life and are less resistant to spoilage than the non-perishable goods. Okay, yeah. While they can be stored for several weeks or a few months under suitable conditions, they are more prone to deterioration over time due to natural processes like moisture loss. I think you did something to do with water activity somewhere in microbiology. If you didn't do, then maybe we'll interest ourselves and do it in our other engagements that would come in. We are looking at moisture loss, sprouting, and also the enzymatic activity. Because enzymes are always available, they just need the optimum temperatures. They need those. I think I don't want to give you. I don't want to sell out something that I want to give you as part of our coursework or quiz. But yeah, we have what we call factors that affect enzyme activity. I hope you know those ones. Okay. So these items typically require proper storage, and we are saying such as cool, dark places. For example, the darkness due to what we could call some of these ones react to light. Okay, and then the light, then it becomes, sometimes they react and they get oxidized. So oxidation takes place. For example, the potatoes, or some are refrigerated. For example, the fruits and vegetables, but still, ideally, you cannot refrigerate them for too long. Still, they will lose their water and they will end up wilting. All right. So now let's look at the perishables. Like the name suggests, here we are looking at the milk, meat, eggs, fish, fruits, and vegetables. We still have the same category of fruits and vegetables in the semi-perishable. However, with perishables, what we have to look at ideally, all the mostly of these animal sources of protein, most of them actually are more perishable. Okay, most of them are perishable. While people could argue that they have higher nutrient contents compared to the other things that are coming from plants, but maybe that is a subject of discussion for another day. That since there are more nutrients in these particular protein foods from animal sources, then they attract microbes easily compared to the other group. However, here we are looking at them having a short shelf life. Short shelf life. With shelf life, I think you will all understand it is storage life, or that particular time it takes for food to go bad. I think you did it somewhere. And we are also saying these are highly susceptible to spoilage. They are typically fresh, unprocessed, and they contain a high moisture content. And this makes them give it an ideal environment for microbial growth and enzymatic reactions. Okay. Yeah. Friends, when we talk of factors that influence enzymatic activity and factors that influence microbial growth, those are very different things. I think we have to take in note on those. Okay. Yeah, however much they may look a little bit similar, but then let's take caution when we are answering such questions when they tend to surface. So without appropriate storage, for example, refrigeration or freezing, we are saying these products can quickly become unsafe to consume. And the perishable goods are often associated with shorter expiration dates, and there is need for temperature control. All right. So microbial presence in raw food. Okay, so here we will be dealing with a few statistics here and there, but really, these are all assumptions, okay, compared to the different microbes, etc. So starting the microbe count, we are saying approximately 10,000 or 1 million microbes per gram. Okay. Yeah. So what are the factors that influence microbe count? If we separated, we could be able to count microbe one by one. We are saying in 1 gram of raw food, we expect to find 10,000 to 1 million microbes. Okay. Yeah. So source environment. Where did you get this particular food? Okay, that will also show that there are a lot of microbes. For example, people tend to tell us that the foods we get from the gardens have a lot of microbes. And also, if we are maybe slaughtering a cow that is where I think they are called abattoirs. Okay, if the sanitation of the abattoir is not optimum or it's not key, then we could have a lot of microbes in that particular meat. And also intrinsic factors of the raw food. The intrinsic factors, those are what we call the internal factors. For example, things to do with pH, things to do with water activity. These will influence the number of microbes that we'll have in that particular raw food. Okay. So handling and processing sanitation, now this is where we come to what we have been looking at always in product development, that once you do not maintain hygiene and sanitation in the place where you're doing the preparations from, where you're doing food handling, or where you're doing packaging and handling storage conditions, etc., we expect you to have microbes that are going to bring a public health concern to the people you're trying to serve or to the people you're trying to sell your product to. Right. Still, when we look at microbial spoilage in vegetables, we are looking at a substrate for yeasts, molds, or bacteria. Okay. Yeah. The microbes, we are looking at different microbes. Okay. So we are saying yeasts and molds or bacteria are present into these vegetables because it acts as a substrate. When you talk of a substrate, what should come to your mind? It could be like a food for enzymes or a food for the microbes in simple terms. So they tend to want to break down these because they feel that they can thrive well in these particular foods because there is abundant of water, there is abundant of nutrients, etc. So bacterial growth in vegetables, and we are also looking at the impact of the intrinsic factors. The intrinsic factors, we have the pH. Okay, I don't know how we could term pH in a softer language, but then we are looking at the acidity or the alkalinity of that particular food. Okay, yeah. And then also we have oxidation reduction potential, which we term as EH. I think at a particular point in time, we will be required to explain a lot on this, but really, there is not a lot that we will want to interest with you right now, but we will talk about it in our later engagements as they come up. Losses due to microbial spoilage in harvested fruits and vegetables. Okay, we tend to understand that whenever you're trying to sell something to someone that has these microbes present, however much you have finished harvesting, then they will reduce on the quality. Once they reduce on the quality, that means even their market value will fall. Once the market value falls of these, that means in the end, you're not going to be making a lot of profit. Actually, sometimes people even fail to break even. And that means they're making losses, even they can't recover the capital that they used to purchase or plant these particular fruits. Okay, so friends, this is basically we are introducing food spoilage, but we'll be talking about most of these in detail, okay? Where we are looking at the gram-positive bacteria like the lactic acid and the leuconos. These scientific names, sometimes even me things go wrong, but it's okay. For the gram-positive bacteria, we are looking at the lactic acid bacteria, and this lactic acid bacteria, or the lactobacillus, is what we find in milk. Okay, when milk is going bad, since milk has lactose. And this is a substrate for the lactobacillus, because lactobacillus, I think, turns that into lactic acid, turns the milk into lactic acid, or that is the end product. Also, we have the streptococci, or some people call it streptococci, etc. Okay. Yeah, so those are the gram-positive bacteria, and in our later engagement, we will understand why these are called gram-positive bacteria, and also we also have gram-negative bacteria elsewhere. All right, and also we are saying the role of staphylococcus in cross-contamination and growth conditions. Here, friends, staphylococcal bacteria is bacteria that is sometimes present in human waste, okay, human waste and also in animal waste. That is why they tell you to always cover the foods that you're not eating, because at the end of the day, if you leave food open, what happens is the bacteria, the vectors that we have, I don't want to call them vectors as such, but these disease transmitting organisms, for example, the flies and also all other insects, the Drosophila, etc. These will go on that particular human waste or animal waste and then come to your food. Okay, yeah, and this will lead to to contamination. Also, this staphylococcus, I think, is also present in some meats. That's why we tell you that you people should not mix raw food with cooked food, or semi-cooked food with raw food. Okay, yeah, there is a lot of cross-contamination that could happen. I think we'll also be looking at that in detail. Well, I know our engagements are always brief. We always have 1 hour. We can't always exhaust everything, but at least I think I will try to, as much as possible, put everything in our slides, that even if we get caught up by time, sometimes you could always go back and revisit these slides and be able to understand a lot. Okay, so influence of high redox potential. Okay, yeah, these are favorable conditions for aerobes and facultative anaerobes. Okay, so here we'll be also looking at the aerobic bacteria. I don't know whether you looked at them. Aerobic meaning these thrive well where there is a presence of oxygen, and then the anaerobes really can thrive even when there is limited oxygen. Yeah, ideally. So we understand for the, if we want to mitigate the microbes that are aerobes, that means we have to reduce the amount of oxygen that we are letting into the environment for that particular storage of that food. But then if it is an anaerobe, then we will have to look at other options to deal with that particular food. Issues to do with the structures of these bacteria. I don't know whether you people looked at them, because we have those bacteria that are thermocytes, etc., which are not affected by heat, etc., etc. So we'll be looking at these in detail later on. Okay, common bacteria spoilage. Here we are looking at soft rot caused by Awinia carotova, or you call it the Pseudomonas species. Okay, that is a common bacterial spoilage that happens to the vegetables. And also we are saying characteristics, the growth is at 4 degrees centigrade and breaks down the pectins, soft and mushy consistency, and there is a possibly a bad odor. Here we could bring a question and tell you maybe detail the common bacteria that is present during the spoilage of vegetables. Really, here you could talk of the Pseudomonas species, and also give us the characteristics that favor its growth. For example, the low degrees, the low temperatures of 4 degrees centigrade, and also it's present in breaking down the pectins. The pectins are the outer covers of the vegetables in most cases, and also I think the fruits have pectin as the external cover. So the softening mechanism, here we are looking at the pectinase producers and lead to sugar fermentation, and also the role of endogenous enzymes in softening of the vegetables. This still will detail further in our engagements. Fungal in vegetables, we are having the grey mold rot, watery soft rot, Sometimes it looks more, how should we say, more knowledgeable for you to detail with us and give us these scientific names, but still you could earn something from me if you give me a name that is not scientific, however much it is these general names. But if it is correct, then you will be scoring maximum points from that. Okay, so microflora in foods, we have looked at the microbes in vegetables, where we looked at the Pseudomonas, we looked at the grey mold, or you call it botrytis, or you call it the Jotrichum, which is called the water soft rot, and also the Rhizopus stolonophylla, something like that, which is the soft rot uh, with the cottony growth, something like that. Okay, so we are done with what we look at when we are looking at the microbes in vegetables. And like I told you, uh, believe me, once you go and do your research and come up with something and then reproduce it in the exam, trust me, I will go and do that research. And once we ascertain that it is correct, you will get your marks. It's not about whatever that is in these slides. That is what is going to earn you maximum marks. Okay, yeah. But if you could go do your own research, then come back and reproduce something, I will go do the same research because that information is available. Do the same research, find it, and if it is true, you will get your marks. So don't worry about anything. Okay. However much I know ChatGPT has come, and I don't know what is the the mode of our assessment, but I know we will work on that because also we should use. I don't detest or I don't prevent students from using AI, okay, because AI gives you more reason to think, but we just want to have your human something into that work. However much you're reproducing AI work, I want to see something of human, and we already see what humans can do and what AI can do, because these days there is plagiarism that catches plagiarism checkers that are capable of identifying work that is produced by these language models like ChatGPT, the ANI, etc. All right, I'm not scaring anyone, hopefully. So the food composition is the typical pH, the water, the carbohydrates, the proteins, this fat content. I'm not so sure about that, but when we talk of palm fruits have oil in them, so that oil is fat. So that is a discussion that we'll have to look at sometime. Microbial dominance, here we are looking more at yeasts, molds, and these are more important than the bacteria. Usually here there are molds and yeasts that attack these particular fruits once they are left in exposure. So here we still, with the yeast mold interaction, we are saying that the yeasts are the first to develop, and then the molds come in as spoilage proceeds. Okay, so specific spoilage organisms in fruits, we have the Penicillium species. Okay, Penicillium, I think you know something to do with penicillin and what it treats. Okay, it is this is acquired, then it is used on bacteria. Penicillin is an antibiotic. Hopefully, we all know that. Okay, so we have still this botrytis cinerea. We have the Rhizopus stolonifer, and then the Aspergillus niger. Okay, yeah. So like I said, you could get a few of these examples when required. You interest us and present them to us. All right. So for microbial flora of beef, we are looking at the gram-negative or the aerobrods. Okay. Here we are looking at the Pseudomonas, we are looking at the Acinetobacter and Moraxella. So for the gram-negative, really we want you to be able to give us these characteristics and saying that these bacteria are rod-shaped with a double cell membrane. I hope we know what the structure of a cell looks like. And we are saying these are aerob, which means they require oxygen for growth. And some strains like the Pseudomonas are known for antibiotic resistance. For example, with these bacteria, even if you give antibiotics, these bacteria are still present. Yeah. If time allows, we'll also be looking at some immunology component, whereby we'll be looking at possibly uh, the broad spectrum antibiotics and narrow spectrum antibiotics, something like that. We'll be interesting ourselves with that. And what is a habitat? We are saying these are found in diverse environments, which include the soil, water, and healthcare settings. And we are saying the Pseudomonas is more commonly associated with water sources. Okay, and we are saying some species can cause opportunistic infections to immunocompromised individuals. Okay, and we are saying in particular Pseudomonas is notorious in causing pneumonia and the urinary tract infections. So you people who usually leave stagnant water around your homes, it's not only the malaria that you will get from the mosquitoes that will be laying eggs in that water, but you're also putting yourselves at risk of getting pneumonia from bacteria that are present in that water that is stagnant. Right. Also, let's continue and look at Bacillus and Clostridium. These are species. We have what we call a Lactobacillus, but we also have this Clostridium. I think it is Clostridium botulinum and also Clostridium perfringens. Okay, and we are saying these bacteria form spores, and these are the resistant structures that allow them to survive in adverse conditions. When we talk of adverse conditions, we are meaning harsh conditions, and we are saying these are typically rod-shaped. So these are found in soil and also other natural environments, and these species, with some of these species utilized in industrial process in medical relevance. Okay, so the bacillus species can lead to foodborne illnesses when spores survive and multiply in properly stored or reheated food. Okay, yeah, that is why we also need to, as much as possible, not eat all the tinned foods we find, because we found them in a supermarket or etc. Sometimes most of these microbes have the opportunity to thrive in there. So Clostridia, like Clostridium perfringens, can cause food poisoning, and also the gangrene gas, and with these various infections, and these are very deadly microbes once ingested or taken in. Okay, they play a role in food fermentation and also in antimicrobial production. Really, that is what we have looked at when we looked at the microflora of beef, the gram-negative anaerobes, the Pseudomonas, CTC, and then we have also been able to look at the Bacillus and the Clostridium species and also all their characteristics. Okay, so the spoilage characteristics in beef, we are looking at off orders due to a short chain fatty acids and volatile compounds. Really, you can always tell when meat goes bad. Okay, yeah, because we will get that off flavor and also slime accumulation from bacterial cells. When bacteria die, I think this is the byproduct or the end product will be something like mucus like, and then in the end we'll be having this slime accumulation. Discoloration due to pigment alterations, then also greening caused by microbial production of the hydrogen peroxide and the hydrogen sulfide. Okay, yeah. So greening here we are looking at still coloration. Okay, the green color that starts to develop on beef due to the production of those particular gases. Also, interaction with the nitroporphyrin compound, etc. Okay, so the microflora of ground meat, we are looking at increased microbial loads in ground meat, the surface really these particular foods. Okay, so the importance of surface conditions in spoilage. Why do microbes require a huge surface area to act on and etc? Okay, so the common spoilage in processed meats, we are looking at the slimy spoilage, hydrogen peroxide, and also the hydrogen sulfide. and the hydrogen sulfide production. Spoilage. Okay. Yeah, here we basically, I think we should have looked at this in food product development, but we are having first semester will have to make sure once you go into your next year, you will always have something that you can able stand in the crowd. We're looking at the typical composition of poultry, 75% water, zero carbohydrates, because most of our animal sources of food or animal sources of protein don't have carbohydrates is because glycogen is the storage form of carbohydrate in animals. Okay, yeah. So once you cannot, most of that glycogen turns into lactic acid once we are slaughtering a cow, or once we are slaughtering a hen, it by the in form of glucose, etc. All right, we are looking at 18% protein and 3% fat, and then the pH is neutral. Okay, so let's proceed to bacterial spoilage. Here we are saying that poultry primary, here we are saying that poultry primarily experiences bacterial spoilage. Okay, so internal proportions are usually sterile. Sterile means that they are capable of they are is not affected, or it is not. Yes, please. Someone is making some feedback. We was still using something to show you sterile that it doesn't have any microbes. Spoilage flora introduced during processing and handling. So during this mishandling is when to bring the food is sterile. And we are saying what does the 80 bacteria. A graphical illustration of the bacterial count increase over time, and also we'll be having, I think, a graph showing bacteria growth. Okay, yeah, but still, we have spoilage. We are looking at the correlation between microflora groups. We are looking at off odors or off flavors that develop fast, and these followed by the visible slime production. So for advanced spoilage, we are looking at the skin pseudomonas or pseudomonads. These we could only see them under ultraviolet light. That is why I was telling that in this session or in this subject of microbiology, we have a lot to cover. Okay? Because the people who are in this field are people who are doing research. You're doing research on very small things that people tend to overlook, but you will not overlook. That is why we always need to wash our plates before we. Remember when I was still in school, in elementary level, basically primary level, whereby you don't have to wash. In most cases, you don't wash the plate with soap. Okay, you just rinse a bit and go and eat. But by God, we expect you to know that after eating on a plate, microbes will accumulate on that food, on that plate. Sorry, and once you don't wash with soap, or once you don't wash with a sterilizer or hot water, it is you go and put other food on that. We are looking at cross contamination happening, and taking care of you. Cases we are looking at Acinetobacter and alkalinogens, Aeromonas and Alteromonas and Moraxellea. So here we could come up with so friends, let us mechanisms. Okay, yeah. So most of these foods, like we talked of, first of all, the egg. Still, we understand that it has protection mechanisms that prevent microbial growth or microbial entry into the. So typical composition of eggs, we are looking at 90% water, then we are looking at proteins, and then a fat of 5% and greater than 9.0 pH. Okay, that means it is alkaline. The neutral is around 7.0 pH. Okay, so the more it goes high in 654, it is going to be acidic side. Yeah, we'll look at pH also, don't worry. All right, egg composition and protection shell, the membranes, the lysosomes. The lysosome is a protective protein that is present in this. Is a God amazing. Okay, and also at the high pH, like uh, alkalinity. So the bacteria that do not thrive well in highly alkaline environments won't attempt to enter the egg and cause spoilage quickly due to the environment where they are in. So for fish and we are having for molasses something of 3 to 5% or 0.5% for the cross-starch chains, 24% of proteins, etc., and 1% fat, and 6 to neutral, though it is more inclined on the acidic side. The high protein content, we are saying this makes a fish a source of nitrogen. I hope you know, because we could be talking of an amino acid looks like. I hope you know. But if we don't know still, it would be good you to tell it to me so that the next time we have you guys never pass first-hand information. That is those. Really, do I want to time? I could watch it when it is happening. For example, right now in class we have Clara. I could interact with Clara right now and George because they are present in class, and for anything I could give them feedback there and then. But when people are not present, then well, at the end of the day, you people are paying. You're paying your hard-earned money, so you should make an effort to come and always be present for these classes. Finally, let's look at the common causes of spoilage. We are looking at autolysis. Lysis is breakdown, really. Okay, so autolysis means things being able to break down their own oxidation, and also microbes being present. Well, the factors that influence this microbial accumulation in fish or microbial quality, we are looking at the quality of water from which the fish was harvested. If the water had a lot of microbes in it, then that means the fish is automatically going to have a lot of microbes too. And also, we are looking at clean water results into fewer spoilage bacteria. The impact of sanitized processing steps and also fresh versus the frozen fish microbial counts. I think that is something to interest ourselves with. For example, with fresh fish, we expect to have, a lot of microbes coming in. But for the frozen, remember now the microbes are inactive. They are not denatured. It is heat, but denatures microbes. But then when freezing just inactivates. So they are inside, but they are not reproducing. So their numbers are not increasing, right? Yeah. Okay.